It is a real honor to be here speaking to you today, and I'm excited to talk about some of the work that my students and I have been working on over the past, well, maybe slightly longer than I would care to admit. I was actually so excited for this that I was a little bit indecisive on the title. So if you saw this one advertised, I'm talking about heat waves also. So if you only came for the heat waves, don't panic. We'll get to that. But before I get to any of those things, I want to thank my lab members past and present, many of whom I'm super happy to see here. So there's several generations worth of students I've gotten to collaborate with. And then the bottom right is my family, who are not only very permissive of me disappearing, at every full moon to go out on the low tides, but actually come with me. So those of you who went on the Stanley Park field trip today got to meet my kid Zoe, who helped us find some of the cool stuff on the shore in Stanley Park. All right. So I work in places that look like this at low tide, and you have these really striking patterns where up at the top, that sort of tan zone is barnacles, and then there's a seaweed zone, and then the gray in the middle is a mussel bed, and then there's kelps and other seaweeds, and then the bottom green bathtub ring is surf grass. And those types of striking patterns are one of the things that makes intertidal shores a really terrific laboratory for studying ecology, because that gradient from high on the shore when you're out of the water and have to deal with really harsh stress down to the very bottom of the shore where you're underwater most of the time but threatened by your natural enemies allows you to understand things. And you can also just turn something over and see what it's eating, which is way easier to do with a starfish than it is to do with a wolf. So the question I'm going to pose here is not original to me. This goes back quite a long time. And that is, why do this many species live here? So we can see, maybe about 7 obvious zones in there. Why is it 7 and not 100 or just 1? So if we take the mussels in the middle, let's see if this shows up. No, all right, the mussels are in the middle. Why don't they just overgrow everything else and we have nothing but mussel bed? And this is the question that was asked 50 plus years ago by Bob Payne. So this is a textbook example. If you've taken an ecology course, you've probably been exposed to this idea. For the nerds, this is the reason that we have this many species here. It is local species diversity is directly related to the efficiency with which predators prevent the monopolization of the major environmental requisites by one species. For the non-nerds, what this means is that if you have something like mussels that would potentially overgrow everything and monopolize space, the reason that it's not only mussels as far as the eye can see is there is a predator that eats mussels and basically saves the day for other things. And that predator is the sea star piesaster. So the photo there is Bob Payne. Sitting next to him is a giant pile of piesaster. And so he did an experiment where he would pull them up and remove them. So here's a picture of a piesaster eating a mussel. And if you came on the field trip today, we talked a little bit about how this works. But basically, a starfish has a whole bunch of little tube feet under its arms. It crawls along with those until it finds a mussel, gives it a nice friendly hug, and then pulls it open and turns its stomach inside out and digests the mussel. So that's what's happening to the one it is clutching. The one just below it was probably breakfast. It's now on to lunch. And piesaster can be pretty devastating predators. They really like to eat an awful lot of mussels. And because they prevent the mussels from moving down the shore, Bob's field site was called the Glacier because of the rumbling, inexorable downward trajectory of the mussel bed. They allow all those other species in the left-hand image, the kelps and the sponges and the big low-shore barnacles, to persist and not be a big mussel bed. Okay, so here we go. Bob Payne's most famous discovery summed up is when you have piesaster, the sea star, present, you end up with about 15 species of the big things that need primary space. And when you don't have Pisaster, you drop from 15 down to about 8. And that's because the mussels have outcompeted those other 7. Okay, so that's what is in the textbook, and Pisaster is treated as a hero, and everybody, throws parades and parties for Pisaster and all this. But somewhat more recently, the people who study the things that live in the mussel bed have lodged a complaint against this paradigm. And that complaint is this: mussels are excellent habitat for all the little worms and sea cucumbers and crustaceans and other things that need those sort of damp, cool microhabitats. And if you're patient, you can identify well over 100 species in a mussel bed. So if mussels provide habitat for 100 species, 100 is more than 15, and so Pisaster maybe is the villain and not the hero. So which is it, right? This is a question that I found very interesting, because the people that are arguing about this were comparing 15 versus 8 and then 100 versus 15. But if you're sort of a logician, you've noticed the error here. It really should be the 8 that have mussels plus those 100, and then 15 that don't have mussels but presumably have something else that nobody else has bothered to count. So there was an opportunity to understand the system better. And so that's what a few of us set out to do. And we were very lucky to do this up on Calvert Island on the central coast of British Columbia. This work was supported by the Hackeye Institute that has their field station there. And you can sort of see the landscape there has sandy beaches, and those are intersected by rocky headlands. And that makes for a really nice study system, because you sort of have these repeated headland units upon which the Pisaster mussel system exists. And here's a photo of one of those. And I don't have a pointer, so I'll try to guide your eye a little bit. You can sort of see my shadow on the right for scale. If you look all the way to the left, that's up the beach towards the forest. And you see the dark bit on the rock way over on the left-hand side? That's a mussel bed. And then as you come down shore, the beach is getting lower and lower and lower as you're getting towards low tide. And then out at the toe on the right-hand side, there's no mussel bed over there. The reason for that is that Pisaster requires a low shore rocky refuge to hang out at low tide. And then when the tide comes in, it crawls up the side of this wall and eats whatever it can get. To the left of the image, you don't have that low shore refuge, and so there's no Pisaster, and that allows there to be mussels. I was very excited to notice this pattern because, thinking about this question, when Bob Payne originally did his work, those experiments lasted between 17 and 25 years. And I'm not inherently that patient, but he would pull up the Pisaster from one area and walk them over around the corner and then hurl them into the ocean. And that was the literal verb he used in his paper. And did that for year after year after year, and that's how you got that sort of glacier of mussel bed rumbling down. Okay, so we have little pre-made, Pisaster manipulations here. So this is a paper that I'm slowly writing up right now with the help of Amelia Hesketh, who's here and created this lovely image. This is the schematic that sort of gives you the general idea. Where the sand is low, you get Pisaster at low tide, and that's the sort of filled stars down there. And then when the tide comes in, they forage up the shore and they push the lower edge of the mussel bed up, or they eliminate it entirely. So that gives us an opportunity to compare the diversity in the areas that don't have mussels to the areas that do have mussels, all at the same shore level, thanks to something that's happening much lower down on the shore. All right, so you probably have been to talks where somebody, relatively senior gives you a cartoon and says, all right, just believe me, that's the way it is. No, don't trust senior scientists. Here's some data to support that. So on the left, where the sand level is higher, you can see there's very few Pisaster in a meter of shoreline there, and lower down you would typically run into 5 or 6 in a linear meter of shore. At some sites less, at some sites a lot more. We have auto-advanced to the next thing, which is you can transplant mussels out like a little buffet for the sea stars. And this is going to be exciting if this auto advances the whole time. All right. I will try to keep up with it. All right, so mussels not eaten on the left. That's what the outplants look like after they have been eaten. And maybe if this is on like a 15-second timer, I can just wait. And that determines how vertically wide the mussel bed is. So this bar graph is upside down only because the top edge of the mussel bed, if it exists on both ends of the headland, is in the same place, and it's the lower edge that's different. So where you have sand higher on the shore and no piesaster, the mussel bed covers more of the vertical extent of the shoreline. All right, so that allows us to sample the mussely areas on one side and the non-mussely areas further down. So we can either put down little quadrats well, actually, we put down 20 by 20 centimeter quadrats, and we dug up everything in there and brought it back to see what was in there. So on the right, that is the no mussel bed area, and on the left is the mussel bed area. And it's kind of hard to explain what's going on in that image. That's Chris Neufeld, who is a normal human with a regular-sized head. And he has put it in, so the video hopefully helps, right? So these mussel beds are non-trivial. You can get your entire head in there, and if you really want to experience the intertidal ecosystem, I recommend this because you can hear the little things happening in there that you normally wouldn't pay attention to. All right, so as much as I was a fan of the whole Bob Payne paradigm, piesaster as a hero, I was starting to think that the detractors are probably going to be right, because that is a bodacious habitat. Incidentally, when Chris pulled his head out, there was a sea cucumber stuck behind his ear. So he's the only person I know that has collected a sea cucumber with his ear. But in addition to cucumbers, there's all kinds of other wonderful things. Some of these species came from the mussel beds during this collection. Others are just things that you can find up on Calvert Island. And there were a total of 135 species in our samples. Now, I'm not great with taxonomy, so it was very fortunate that there was a bioblitz occurring while we were doing some of this work. So this is another opportunity to thank Hackeye, who is supporting the bioblitz. So, I can do some of the snails, but when it comes to worms, I'm pretty helpless. So we would, bring our sample in, we'd sort it out into all these different piles of animals that look similar, and then I would carry my little petri dish of worms up to the Leslie, the worm expert. And she was very excited to see me because her love for worms is so deep. And so she would explain, all right, this is this species, and this is this species, and this is this species, and I'm writing notes, and then she would say, but these are spionids, and I don't do spionids. And my heart would break a little bit. And then she would say, so you will have to talk to Vasily. And the guy at the next microscope would be like, hello, I'm Vasily. And I'm like, this is the best. Anyway, thanks to the hard work of all these taxonomists, we got really detailed information on everything that was bigger than about 2 millimeters in these samples. And here is what the first two sort of headlands looked like, right? There were 2 samples in the area without piesaster, 2 samples in the area with piesaster, and the mussel bed really did look like the richer habitat. And I was like, Bob Payne was my PhD advisor. I was kind of sad for the paradigm. But we had 5 samples, and the other ones showed different patterns. In some cases, there was no real difference, and for 2, for West Beach and 7th Beach, there were actually more species in the areas where piesaster was foraging, and there were no mussels. And that came as a bit of a surprise. So we realized it was important to consider not just whether it was mussels or not, but what was replacing the mussels if they weren't there. So now I've color-coded things by the other types of species that were around that piesaster leaves behind. So pictured are piesaster down in a sort of low-shore crevice. Then all those thatched barnacles there, they are happy to walk over those to get up higher and get the mussels, because the mussels either taste better to them or are more energetically profitable or something. So by leaving the thatched barnacles there, there's still a lot of habitat. And the thatched barnacles are those yellow bars, and it turned out they were terrific habitat for all the little species that live there. And I was so stunned by that. You remember that mussel bed with Chris Neufeld's head in it? Thatched barnacle habitat is only like 3 centimeters tall. And I knew that there's some stuff. There's a shore crab up there and a couple of whelks and some limpets and things. So I didn't trust that result. So we went back out and found other areas that were sort of a patchwork quilt of mussels and thatch barnacles and sampled just those. And sure enough, thatch barnacles, kick-ass habitat, more species than in the mussel bed. Who knew? Amelia eventually found this to be very true in areas around Victoria as well. Okay, so maybe Bob and his ideas are rescued because Piesaster, by eliminating mussels, yeah, there's things that live in the mussel bed that are sad, but there's more things that live in the barnacle bed. But there's a trick here. It turns out that the species list in a barnacle bed is sort of like the menu at Tim Hortons, which if you're not from Canada, that's the famous donut and coffee shop. There's a reasonably long list of donut types and soups and salads and sandwiches and wraps and stuff. But if you go to a different Tim Hortons, it's the same list. As opposed to, say, an independent bakery, which might have fewer options. But if you've got a neighborhood with 5 independent bakeries, then you probably have more options than the 5 Tim Hortons would have provided. And that's what we're seeing in thatched barnacle beds. So if you've never seen this type of plot before, the way to read it is the further apart in space the points are, the more different those communities are. So the mix of species is different in some way. So the mussel habitat, you see how far apart those mussel points are? And they're also different than the thatched barnacle points. So those two habitats really do host different groups of species. But all of the thatched barnacle points are pretty close together. So every thatched barnacle bed sample was pretty similar to the next one. You weren't adding very many species at a time. So this is where I realized that my initial question was really naive because I had been thinking at the plot scale. It's like, all right, let's just do what ecologists have done for decades, go out and count things in an area and then average the number of counts across a couple of replicates. You get a number, you compare that number to the other treatment, and then you walk away. And clearly there's more going on because just averages hide this beta diversity, this turnover in diversity from one plot to the next. So back to those original samples across 10 headlands, the mussel habitat did have the most species, and it had a lot of species that were only found in the mussel bed samples. But each of the other habitats, including the little brown barnacles, which are like 5 millimeters across and are really poor habitat, they still had an isopod that wasn't found anywhere else. So really the key here is not whether you have, a mussel bed or not, it's what else is around. And if we do that same sort of analysis we just did, now the mussels here are on the left, so that's the no Piesaster mussel bed thing in the tan. And then the area where Piesaster is foraging, you have all those different community types. Sometimes it's thatched barnacles, sometimes little brown, sometimes seaweeds. You get a wider diversity of communities. So again, we have this sort of, do you want to just measure at the local scale and look at alpha diversity, or do you want to look at the larger scale and see how different each plot is from the next? So to sort of boil this down into a digestible summary, why do this many species live here? Well, first, Bob Payne was right. Predators help maintain a diversity of habitat-forming species. That includes a lot of these seaweeds and barnacles that the mussels would otherwise overgrow. The detractors of that paradigm do have a valid point because habitat-forming species support many additional species, but not just the mussels, right? The thatched barnacles, the seaweeds, they all are spectacular habitat in their own ways. And it's this landscape scale pattern of whether there's the low shore refuge for the predator or not, which determines whether you have mussels. And then there's some additional factors like how wave exposed is it that determine whether it was little barnacles or big barnacles or seaweeds at these spots. So that landscape scale sort of pattern drives how this balance plays out. So really, if you want the most species, it's not, yes Piesaster or no Piesaster, but both. And that sort of helped me see the system in a much more nuanced way. Okay, so as with the forest, the biodiversity is all about balance. All right, so by quick show of hands, how many people feel that the planet is in balance right now. That's cause for optimism. I don't necessarily feel that way just because I'm a climate change ecologist. And there was just a headline on the BBC website 2 or 3 weeks ago that was talking about the fact that 2023 was the warmest year for the ocean ever recorded. And they were interviewing a British ecologist named Nova Mieszkowska, who happens to be a friend of mine. So I was excited to see her in this interview. So you can probably still find this. And my personal favorite part of the interview, I don't know if you can read the tiny words at the bottom of the screen, her quote was, "If I were a barnacle, I'd be quite worried." So people are, well, me and Nova are thinking about the barnacles and what it means. And we've known this for a while, right? It's not just that 2023 happened and we were like, oh shoot, we need to start paying attention to temperature. This data set here is the Victoria airport air temperature record just for like summer daytime highs back to 1950. So this is not new. Climate change isn't, it's often framed in the media of like, oh, our children are going to have to be dealing with this. We are the children of people, so that statement could have been true for our parents and also for our grandparents. So since the 50s and during some of the sampling for this project, I was staying in a motel in Victoria that was just brutally hot because it didn't have air conditioning. And the clerk at the front was like, well, yeah, when it was built in 1950, we didn't need it. So we know that it's getting hotter, and we know that ecosystems are responding. And you remember that wonderful zonation pattern in one of the early slides? That is one of the things that we can document change with. So this is a paper that's now 13, 14 years old. But it compares data on how high up on the shore the barnacles and mussels live. That's the triangles and open circles there, where the lower limit of the mussel bed was. And the original data were collected by a UBC master's student in the 1950s. There's a nice big wide mussel bed. And then when I went out and redid this in 2009 to 2010, that mussel bed has shrunk. So with the headland example, you see the bottom edge of the mussel bed moving depending on what piesaster are doing. In this case, there's piesaster everywhere, whether it's 50 years ago or more recently. It's the upper edge of things are being forced down because the hotter it gets, the less time you can spend out of water. So a barnacle or a mussel needs to be lower on the shore to avoid fatally overheating. Okay, so that is a bit out of date, and fortunately we have Rebecca Hansen, who I think is going to be around. I'm not sure if she's here yet, but she did her master's looking at updating these patterns and doing some more mechanistic experiments to explore what set some of the upper limits. And she found that the upper limit of barnacles has continued to go down, the upper limit of mussels has continued to go down, and the lower limit of the mussel bed, well, that's actually done something different than we were expecting. That is almost certainly due to the outbreak of sea star wasting disease, which happened about 10 years ago. This is the same pattern of the lower edge of the mussel bed extending down has been seen in Washington State and in Oregon, so it seems to be fairly general. But I think it is probably transient. It might be transient at the scale of 20 years, I'm not sure. But we'll see how long that reprieve for the mussel bed works. But I show you this not for the sake of talking about sea star wasting disease, which could be an entire lecture on its own, but to sort of set up this idea that a lot of our understanding of how ecosystems change come from graphs like this, where there's only 3 time points. And that doesn't allow us to really explore what some of the detailed mechanisms might have been. Was there a specific heat wave in there? If you didn't know about sea star wasting and when that happened, that lower limit thing might be a mystery. So every once in a while, you do have a data set like this one, which was collected by Bob Payne, where it's the upper limit of a seaweed turf. And boy, credit to Bob for collecting the most boring data for 15 years where absolutely nothing was happening. But then there were a series of hot days that corresponded with almost flat calm seas, and that left this seaweed high and dry. So you see a first sort of disruption to its upper limit in 1993, and then 1994, and then 1995, and that last one was severe enough to reset it. And then the whole that happened again in 2004. And so because Bob was collecting these data every 2 weeks in the summertime, that's what allowed us to find the heat wave or the weather event that actually created these patterns of change through time. So that leads me to one of the better quotes available in climate science from Deke Arndt, who works for the National Oceanographic and Atmospheric Administration of the US, and is paraphrased here: "Climate trains the boxer, but weather throws the punches." So we are now living in a climate that is different than the climate when I was a kid. But it's, climate per se isn't killing mussels or seaweeds. It's these individual weather events that are the punches, the heat waves. So this is something that my students and I have been thinking a lot about and trying to sort out, what some of the ways that the coastal ecosystems might be responding. So, for example, Amelia has put out black and white tiles. These heat up passively in the sun. She did this over 2 summers, so we can compare what happens if you get one hot summer versus consecutive hot summers. If you want to know, go see Amelia's poster. There's other things you can do with those tiles. Another student named Cassandra Konechny, along with Graham Brownlee, figured out how to heat tide pools using a propane turkey fryer as the heat source. So I proudly present the Seaside Array for Understanding Thermal Effects, or SAUTÉ, and I do not apologize for that acronym. So we can do things like that. Sandra Emery, who will be presenting a talk not on this, but she'll be around, so you can ask her about this, took propane camp heaters out and then hung them upside down over the rockweed beds so you could look at local adaptation in rockweeds. And this last one, and these are like well-designed, beautifully curated experiments, and the students put an incredible amount of effort into these. And for this one in particular, it was a little bit tragic in that Sandra was trying to heat the seaweed bed up to about 35 degrees, which is stressful for this species of seaweed. And she designed it to have, one heating event that was earlier in the summer and one that was a little bit later, and had all these beautiful multiple stressor hypotheses worked out, and did the first heating event. And then the day she was scheduled to go out and do the second one, we had the 2021 heat dome. And if you weren't in Western Canada or the Pacific Northwest in the US, it was really something. These are sort of like the atmospheric modelers put this together. This is what the temperature patterns looked like on the sort of hottest day of the heat dome, which was June 28th of 2021. And if you can't read that scale bar, these are deviations in temperature in degrees Celsius from typical, and it goes up into the 20s. 20-something degrees hotter than typical is ludicrous. And there were all kinds of effects of this heat wave. The star there indicates where you can find the town of Lytton, which I think it was on a Sunday, it broke the all-time Canadian high temperature record. On Monday, it broke it again. On Tuesday, it broke it one more time by, almost 5 degrees more than the, record had been before this heat event. And then on the following day, it burned down in a fire that may have been sparked by a freight train, and 2 people died. So it was the kind of thing where if you could put it into a movie, people would be like, no, that's a little too over the top. Really incredible event, and lots of ecological implications of this. And if you want to hear about some more of the ones that aren't just marine, Julia Baum and Michelle Sang and I have just figured out how that we're going to ad-lib tag team our presentation on that topic on Tuesday afternoon. So you can come and learn about how the bees and the birds and the trees all fared. All right, so what did the shoreline look like? Well, here we are in Lighthouse Park over in West Vancouver on the hottest day of the heat wave. And it's kind of hard to see how hot it is, but the undergrad and I who were out there were actually a little bit worried about our safety. It was like being in an oven, so we would go out and like take some pictures and then retreat into the shade and drink water and eat frozen grapes and oh, okay, and then back out. And we had a thermal imaging camera with us. So this is what the shoreline looked like to the eyes of that camera. The way to read this, the number on the upper left just shows you what the little crosshairs in the middle are pointing at, which is the water in this case. So 23.2 is not especially dangerous. The scale bar on the right shows you the coolest and the hottest parts of the image. So pay attention to the number in the upper right. Some of the rocks were getting up to 52.4 degrees C. And the mussel bed got up as high as 56.7. And we didn't take very many of these pictures because it was like, man, let's get some data and get out of here. So I'm sure we could have found hotter areas. Coincidentally, the hottest day recorded by a weather station on Earth is also 56.7 degrees. This is way above the tolerance limit of a mussel. And if your eyes are sharp, even though this is a pixelated image, you may be able to tell that those mussels are gaped open because they had already died by the time we were there to take these pictures. Lots of things suffered because these extreme air temperatures happened on sunny days with really low tides, and those low tides were kind of like early afternoons, like the worst time of day to do it. So we found some sea stars that had, anything from healthy to sunburned to deceased. There were areas where shallow water behind a sandbar had, lots of dead kelp crabs in it. There were whelks and shore crabs. Lots and lots of things got unlucky. But those examples are all mobile species. And invertebrates are kind of smarter than you might imagine. So if they get a little bit of hot weather, they use that information to go hide. So on the next low tide, they're a little bit more protected. So by and large, animals like our friend Piesaster didn't suffer too badly because they were able to get into the shade of rocks or retreat into deeper water. The species that really had trouble were those that are attached directly to the rock. So things like the rockweeds that Sandra was studying and the mussels and the barnacles. And I can't share with you the smell that this produced, but you could smell it several blocks from the shore. And it was kind of viscerally depressing to do this field work because you go to a site you hadn't been yet and you're sort of worried about what it's going to be like, and you get out of the car and you smell the smell, and it was really terrible. So the beach down at, or the boulder field down by Kitsilano Beach with downtown Vancouver in the background, the photo on the left is just after that heat wave where things have died but not yet washed away. And then on the right is the following spring when all those rockweeds are now gone and the mussels on those boulders are gone and most of the barnacles on those boulders are gone. And yeah, you would just have situations like this where it would just be dead mussels for days. There were some cool patterns, though, that emerged from this, so we did learn some useful things. One of those is that there's actually a really important role of local scale topography. So looking at these boulders, these things have, flat surfaces and steep walls, and parts of them face north and parts of them face south. And that has a really strong effect on how much heat, say, a mussel would experience. Because if you're on the south side and in the sun, you're going to heat up much more than something in the shade on the north side. So we went out and measured that and produced graphs like this for a number of species and a number of sites. So along the x-axis along the bottom, that's the angle of incoming sunlight. 90 degrees means it's just basically, take the sun where it was mid-afternoon, hottest part of the day, and a photon comes down, and if it hits the surface like that, that's that 90-degree, most direct sun you could get. And then moving, around a corner or over a ridge, you're getting smaller and smaller angles until you hit the zero line, and then anything past that, you're in the shade, right? And then we went and just put down little quadrats and quickly estimated what fraction of the mussels had died. So we were sort of the coroner service and doing this as quickly as possible. Mussel beds, and this data set comes from the Stanley Park site that we went to on the field trip today, for those who are curious. Any mussels that were in the sun were really in trouble, and mortality was sort of 90 to 100% until you got right up to the shady, the edge of shade or in shade in the middle of the day. And at that point, you were okay. So important local scale patterns there. There were also larger scale patterns to this mortality event, in part because of where it got hottest. So Vancouver got really hot, Nanaimo got really hot, the whole Strait of Georgia area got fairly hot compared to, say, the outer coast. The other big difference between the Strait of Georgia and the outer coast is the timing of low tide. Because at low tide, let's see, imagine that this is the entrance to the Strait of Juan de Fuca and Vancouver's over there, and I'm low tide. It's going to take me 6 hours to get from Tofino over here to Vancouver. And so the low tides were very early, well, relatively early in the morning on the outer coast when it's not especially hot. But they happened at like 1 or 2 in the afternoon in places like Vancouver, where it was just worst case scenario hot. So that's what those tide traces are indicating. Those vertical lines are solar noon. And you can see Vancouver, those lowest lows are right around solar noon or just after. So as a result of that, we saw much more mortality in the Strait of Georgia than we did further north up on the Central Coast at Calvert Island, or even west out towards Bamfield, out the Strait of Juan de Fuca. And you don't have to know what all those different species are, but of those wee little pie charts, the more black there is, the worse it was. So around Vancouver, there were areas with 70, 80 or higher percent mortality. That's also true up further in. But once you get down to Victoria, it wasn't nearly so bad. The tide was a little earlier, and then further west, the mortality was pretty negligible. Another thing that was interesting about this was who died. So our mussels and our barnacles, which I consider pretty thermally tolerant, really suffered. But there were a lot of species that were introduced from some other part of the world, either accidentally or intentionally, that were by and large fine. So that seagrass there is from Japan. The sea anemone in the next one over is also, I think, Asian. The oyster there is the Pacific oyster, which is cultured here. So if you go, get some for dinner down at Granville Island, that's this species. The mud snail, the sponges, the clams, the limpets down along the bottom, all of those things originated in warmer parts of the world and were transported here by shipping or aquaculture. And so they, in an evolutionary sense, are pre-adapted to events like that heat dome. So we're starting to see a potential shift where we're losing the natives. So there's a pile of dead and empty mussels below a rock that's covered in mostly dead barnacles, but all the oysters on that rock are alive. So what are we going to see with this sort of replacement driven by some of these climate effects combined with invasion processes? And the oysters, I have to say, the Pacific oysters really taking off in the Salish Sea now. Not only are they potentially benefiting indirectly from events that would kill off some of their competitors, the water is finally warm enough for them to spawn in a lot of different parts of the province, and they are taking advantage of that. And so you're seeing beaches like this one that are much more covered with oysters. All right, so we're worried about that, right? Because the native species that, like the mussel bed, co-evolved with mussels. They did not co-evolve with this species of oyster. So another current PhD student of mine, Natalie Rivlin, is working on this, and unfortunately she couldn't be here, but she sent me data like 2 hours ago. So this is sashimi grade data. You are the first people to see it. She has been sampling the diversity in mussel beds versus oyster beds, and the oysters are actually a pretty good habitat for the species we have here. It's not all the same species in the oysters and the mussels, so there's still going to be winners and losers. But in terms of a biodiversity crisis, we will still have biodiversity. And the most exciting thing about Natalie's work is she dragged propane heaters out to simulate a heat wave, and it turns out the oysters were better at resisting that. So in some ways, maybe the oyster invasion is good news. We could debate that philosophically if you want. All right, so is there anything we can do? I once gave a presentation to a UBC biology honors class where I, make jokes and tell stories and give all this depressing news. And one of the students raised her hand and said, "How can you give such an animated talk when all of the news is so bad?" And I said, "Oh, well, you have to sort of compartmentalize the joy of discovery and, love of nature with some of the implications of those discoveries." And another student raised his hand and said, "So you're telling us you're pretty much dead inside." So not only do I not want to leave you with the impression that I'm dead inside, I don't want to leave you with the impression that you should be dead inside either. And there are things we can do. And one of the most inspiring things that happened after the heat dome, because it made the news and billions of animals died and we have numbers to back that up, I got an email from somebody in California who runs a fabric company, and he says, "I know it's too late for this one, but for the next one, do you think it would help if I sent you for free 5,000 lineal yards of fabric that you could build into like pop-up tents and then have an army of volunteers run out and save the poor barnacles and mussels?" That is a brilliant idea. Not necessarily practical at scale, but it would work. And Amelia proved that it would work by having a shade experiment that, like poor Sandra's, was installed before the heat dome hit, and then bam, there was this big catastrophe midway through this experiment. The original design was looking at the thatched barnacle there, which, up on Calvert Island was so important to see what it was doing. So she had areas with and without the barnacles, and then areas with and without these shades. And then the heat dome, again, very instructive. For the areas with no barnacles, the red is the unshaded area and the blue is the shaded area. So putting shades up does work, right? You get more species persisting in those areas. But you know what works just as well as shades? Barnacles. Like those little heroes, they are so underappreciated. But having barnacles around, even if there wasn't a shade, was enough for, the barnacles died, but they still provided enough protection that a lot of the other things were able to persist and even take advantage of the empty shells that the barnacles left behind. So that gives me cause for hope too, because although we're not going to stop global warming as individuals on a realistic timescale, although we should still be doing things that will help, we can take local action like protecting biodiversity. So if we can help maintain the barnacle beds, that's a win for biodiversity, and it buys us some time. And then the last thing, and this is what I'll leave you with, that gives me cause for optimism, is ecosystems really are resilient. So here are the two photos you saw earlier from Kitts Beach up to 2022. By the summer of 2023, that ecosystem is starting to recover. And as of about a month ago, we're now seeing the rockweeds and the mussels back. We're starting to see the species that use those habitats recover into the system as well. So if we can, as people, sort of be on good behavior and help where we can, nature, I think, can take care of the rest. So I'll leave it there, and thank you all for coming.